The title of this lab is the photosynthesis lab. The question is, what factors affect the rate of photosynthesis in living leaves? Hypothesis. If the source and intensity of the light is changed from sunlight to pink light, then the rate of photosynthesis will slow down in the living leaves. Materials. Baking soda, 100 milliliters of water, soap, living spinach leaves, a hole puncher, a beaker, a phone for timing and taking a video, a syringe, pink light, and sunlight. For this lab, we had to make the leaf chads sink. To do this, we took a syringe full of the chads and the solution and created a vacuum. To create this vacuum, we put our thumb over the syringe and pulled down while shaking. This takes the air out of the leaves and replace it with water, making the chads sink. We then place the chads into the beaker with the rest of the solution, and then we can carry on the rest of our experiment. Variables. The independent variable in this lab was the light source and the color of that light source. We were changing the light source and the color in order to test if the color of light sources will affect the rate of photosynthesis. In this lab, we first tested the effect of regular sunlight on living leaves. Then we tested the effects of pink light on the living leaves. The dependent variable of this lab was the rate of photosynthesis. By changing the light source, the rate of photosynthesis in the leaves are then changed depending on the light. This shows that light affects the rate of photosynthesis, and if the light is changed, then the rate of photosynthesis is then changed as well. The constants are what stayed the same in the lab. In this lab, the water, soap, baking soda solution, and the chads of spinach stayed the same. These are the factors that did not change, and then we experimented on them with the other factors.
The data table is showing the rate of photosynthesis for the pink light was slower than the rate of photosynthesis in the sunlight. It took a total of 3 minutes for the chads in the sunlight to float. It took a total of 13 minutes for the chads in the pink light to float. The data analysis showed that the rate of photosynthesis in the sunlight was faster than the rate of photosynthesis in the pink light. In the sunlight, all of the chads floated in the span of within 3 minutes. In the first 5 minutes, 5 chads floated. In the second minute, 4 chads floated. In the third minute, 1 chad floated. In the pink light of all the chads floated in the span of 13 minutes. In the first 3 minutes, none of the chads floated. In the fourth minute, 1 chad was floating. In the fifth minute, 4 chads joined that one chad that was floating. In the 6 and 7 minute marks, no chads were floating. In both the 8 and 9 minute marks, 2 chads floated in each minute. No chad floated from 10 minutes to 12 minutes. The last chad floated at the 13-minute mark. In conclusion, in this lab we tested the effects of different light sources on the rate of photosynthesis in living spinach leaves. Our hypothesis was correct. When the light source was changed from a high-intensity light of 3.86 times 10 to the 26th power of watts in sunlight to a low-intensity light of 9 watts, which was the pink light, the rate of reaction slowed down. This was due to both the intensity of the light and color. The pink light only held pink light and had a low intensity, slowing the rate which sunlight has all the wavelengths of light, including blue and red, which are the wavelengths photosynthesis prefers. To test this further, the light source could be changed again with the same constants to see the effect of a different color light and intensity on the rate of photosynthesis in the living spinach leaves. The lab showed that the light has a large effect on the rate of photosynthesis and that sunlight is the best source of light of fast rate of photosynthesis.